Hey everyone, I'm Stefanie Kall, Senior Staff Product Manager at Countable Labs. And today I'd like to share with you how Countable PCR overcomes the limitation of other PCR methods for your gene of interest assays. So if you're working with qPCR, you know that in order to get the most accurate quantification of your targets, you also need to run a standard curve, which requires more material and effort. And it also means you're not getting a direct and independent readout of your gene or genes of interest. Samples run without a standard curve give very poor accuracy, which ultimately impacts the confidence in your results. Now, digital PCR is meant to overcome the lack of absolute quantification from qPCR. However, dPCR uses Poisson correction in order to determine the number of molecules that are present in the sample. And statistical methods are powerful but limited because they're still extrapolating certain information. Here we see that because of the Poisson correction, once the target molecule number goes above 100,000 molecules, variability increases dramatically. Also, since Poisson statistics are dependent on having empty partitions, your sample input reaches saturation pretty quickly. That means you need to dilute your samples, which introduces pipetting errors. And it also means that if, for example, you're running a single sample and trying to probe multiple target genes at once, and they're present at different amounts, you're going to have to run multiple dilutions of that sample. And the comparison of those different target genes won't be exactly apples to apples. But with countable PCR, we change all that. Countable PCR counts exactly what's in your tube. Since you're amplifying and measuring directly in the same sample tube, you can load way more material with a much higher number of target molecules and accurately and precisely quantify just how much of a gene is present. There's no standard curves, no Poisson statistics, just direct counting. So connect today with one of our experts and learn more about how countable PCR overcomes the limitations of Q and digital PCR.